सर कहेंगे ना मुझसे से कहेंगे Good afternoon, everyone. Good afternoon, everyone. I welcome you all to the second technical session. So for this, I would like to request Dr. Pooja Arora ma'am and Dr. Sudhir Bhardwaj sir to please chair and co-chair the session. Please, sir and ma'am. Working as a data principal in Global Research Group of Institutions in Pharmacy Department. Today I feel honored to introduce Dr. Ranveer Dahiya, our next speaker. Dr. Ranveer Singh is working as an assistant professor in Department of Pharmacology, Central University of Punjab, Bhatinda. Today, the topic of his talk is in silico tools as alternative to animal studies. He has many awards in his credit, like Saroj V. N. Sharma Award, APP Best Faculty Award. He has guided 10 PhD scholars, 10 M.Pharm students, and 10 PharmD students. He is the author of 9 books with a high impact factor. He has 100 national and international publication in his credit. He is expertise in his handling of biopack 4-channel data acquisition system. He has experience in handling semi-auto-analyzer, student crystallograph, electroconvertometer, techno-rotarod, actophotometer, and portrait. Experience in handling spectroscopic and chromatographic techniques. He has expertise in vitro and in vivo screening methods for evaluation of anti-hypertensive, anti-inflammatory, anti-diabetic, diuretic, neuromuscular blocking agents, and central nervous system active compounds. That was a brief introduction of Sir. So please welcome on the dais.
In good time, you are already you are born and as well as artificial intelligence. Now, if we are in the pharmaceutical field or in the medicinal field, this is the primary thing that we should have some molecule or we should have some process and discovery process. But if we talk about the drug discovery or the drug development, that itself is a very small. When we talk about the post drug discovery, it's a very lengthy process. If we talk about post, that is in the billions of dollars. When we drug discovery process, we talk about what should be the methods, how we can go the drug discovery process. So that's why there are so many components of the drug discovery. If you have synthesized any molecule, but you want to find out the potential, the biological potential of that molecule, you should have some methods how you can evaluate. So my topic is about these methods, how we can evaluate these models. So briefly, we can say that as the topic of my talk is alternative to animal models. Animal models are considered that before going into any studies, if your compound, if your molecule is having some effect, it is having some efficacy, then only you can shift for the human trial. But before that, we are going with the animal studies. But nowadays, a different thoughts are thinking that when you are using the animals, why we should not use the animals. So this is a conventional method. Simple any formulation you have tested or any molecule you have discovered, you will then finally be used for the human consumption. Considering these factors, that there are certain factors we should avoid animals. So there are alternative methods which we can opt out. Just like this, there are so many alternative methods these days which are being used or which are being utilized. So I will just try to give you the glimpse of these models which can be used as an alternative to the animal testing. So the first thing is about the use of animals. If we talk about the use of animal into the biomedical research, lots of animals are being used throughout the world. And this is a simple stats. That these are the different procedures in which we utilize the animals. For example, there is a research to understand the body and health, because we say that generally small animals which we are using in our lab, they are having the similar physiology. So that is not anatomical similarities also. That's why just understand the human nature, human physiology, human disease. We use the animal. Second, medicine. Thanks for the development of new medicine. And finally, the major portion is for the genetically modified animals. Now, this is a little bit new area in which that there are certain diseases, some factors which are not present. So that's why we are going with the genetically modified. A major chunk of animals are being used for this genetically modified animals. But we say that we should go with the animals, we should use the animals. But there are different thoughts of schools which are saying that why we should use animal or why we should not use the animals. So there are three different schools of thought. One is purist, second one is ethicist, last one is the evolutionist. Purists are saying that we should use the animals. And only the use can give you some understanding how the drug will behave if we are using these drugs for the humans. And there are ethicists. They are saying that okay, you can use the animal, but when you are using the animals, these animals are not justified. Means they are not treated humanely. There was why there was a question from one guy in the previous session. He was saying that why we are opting out for the animal, we are opting out for the software. Why we are not using the animals as such? Actually, there are certain conditions which were responsible that we have to withdraw the use of animals into the graduation area. There was a raid in 2010 in the rainbeds, and they were animals. They were using the animals. The dogs were used there, and the conditions, because we are testing our drugs onto the dogs. But the condition of the dogs were just like they were having abrasions, they were having skin infections. Even the animals, they were not treated properly. And the result or the medicine which we are testing on those infected animals, moreover, the condition of the animals was so bad that we cannot say that this is an human act. Another one, there is a lab into the Pune National Immunology Lab. In that condition, there was a monkey. Her name was Paro. And there were certain generation of those animals in those cages. When there was a raid from this, you can say, PETA, the animals were released from the cages. But the animals were so much acclimatized to that environment that they were not able to move out of that cages. They were placed outside the cage, but still they were remaining near the cages. They were jumping around the cages because they have forgotten that what is the natural habitat. They were confined to that place. Means they were not justified. There some animals, the monkeys were having broken teeth, broken nails, and abrasions, and all these things. And moreover, there was a change into the behavior. Means monkeys are wild animals, but still they were into the cages and they were remaining there. And that is the reason why it was considered if you are using the animals into the research, then their use should be justified. They should be treated humanely. And that is the thought from the ethicists. What they are saying is that if you are using the animals, their sufferings, their pain should be minimum. But another thought of school is that which is called as the revolutionist. And what the revolutionists are saying, okay, when you are using the animals, they don't serve any good purpose. Means that what results, what we are getting into the animal, we cannot 100% postulate that will be our results into the humans. And these are some of the reasons why there is a revolutionist thought. Means reason behind it that there are three major areas: sepsis, burns, and trauma, in which the behavior or the pathogenetic behavior of the animal is different. In case of humans, that is the different. So if we are doing any research considering sepsis, burn, and trauma, and that is that we are predicting that we will get similar type of results into the humans. That is not possible. Another one is that toxins, which behave differently when they are into the animals. But their behavior pattern means their efficacy, their effects, side effect, adverse effect, it may be anything. Means that their effect is totally different in case of human use. Another one, for example, I think that all the pharma students are most aware of this drug thalidomide and all. These drugs were given into the humans. These drugs were given for the morning sickness. And when we are saying that when we are using the animals, or when we are introducing any drug molecule, it is first given to the animals, and we find, okay, this is safe, this is not having any toxicity, it is not having any side effect, only then it will be recommended for the human use. But this thalidomide, that was considered quite safe into the animals. Thalidomides, tragedy, every pharma students or every person no pharma background know that. But Focomelia was responsible. Means it was considered safe into the animals, but results were totally different in case of humans. So that's why we should use animals when we are having different results. We cannot 100% postulate our results from the animals to the humans. So that's why there is no use or there is no validity of the animals used for research. And another study by the Dr. Richard Fusner. What he is saying is that the history of cancer research has been a history of curing cancer in mouse. You know that those who are into the research, they know it very well. Any study, any anti-cancer, it might be harmful, it can be of any synthetic reason, and if you are using any research or producing any research, you might have cured each and every type of cancer 100% accurately in case of mouse. No matter which formulation is there, anything is there. We are having 100% cure of cancer if we are studying in case of mouse or laboratory animal. But are we able or are we getting the similar results into the human? Are we able to counter the cancer properly? 100% cure is that is the reason that cancer has been cured in case of mouse properly, but similar results or that accuracy is not observed in case of humans also. Another one example is that there is a connection between the alcohol consumption and cirrhosis. But when we are using the mouse model, we are not able to develop that cirrhosis with the mouse. That will not be developed. And again, some baboons are only having the capability, but the results are not accurate in that. So when we are not creating any disease or the exact pathogenesis or the exact path of the disease is not there, then why use the animals? So that is the major concern about the ribolinists. What they are saying is that we should devise, we should develop, we should have some alternative to the animal use. Sorry. I think the reason behind to have this one alternative. First thing is, you can say that is just because of human nature. We are saying that there should not be any harm to the animals. There should be cruelty product. And now I think that some of you might be knowing that if this product was not tested in case of animals, and like this, some even say cosmetic products. It is being labeled on that product that no animals were harmed for this, and it has not been used into the animals. The reason behind is that we are not being cruel to the animals, or we are not affecting the animals in that. Another option for this is we are having more scientific options which can give a little bit more accurate results. And human tissue toxicity is much more relevant as compared to the animals. Suppose if you are taking any human tissue and you are finding the toxicity result in that. So that result will be much more accurate as compared to those that there is some variation when you are going for some other non-human model. So that is the reason why we are saying that we should opt out for some of the alternative methods. So I'm going to give a brief preview of all these, you can say, alternative to animals models. And these are the alternative to the animal use. First one is your cell culture, human tissue, microorganisms, organ on a chip, in chemical testing, microdosing, volunteer studies, in silico studies, as well as epidemiological studies. So these are some of the alternative we consider that if we opt out for these factors, at least we can reduce the cruelty to the animals and we can save the animals also. So this is the first one, cell culture, and everyone is quite aware of what is the cell, what is the function of a cell, and how is the role of a cell into the body. Means that suppose if I'm using any human cells, means the physiology is same, anatomy is same, functions are totally same. And if I'm testing my compound onto any cell, so can you say that there will be some variations even I'm going to use that compound for the human use? We will get the same results. That's why this is the simple method that you can grow the cell into a culture. It's easy process also to grow the cell into a culture into the laboratory conditions, so we can use the cell into that, and an easy process. And you can use these drugs for the evaluation of, you can say, some of the toxicity as well as efficacy for the cytotoxicity also. So that's why growing cells into a lab is quite easy. Moreover, you will get a bit more relevant result as compared to known being used. So this is the procedure how we are doing it, that you can have some cell inoculation and centrifuge. You can make the colonies of your selected cell, and after that, that selected cells can be used for the further evaluation of your compound. It can be anything. You want to study the toxicity, cytotoxicity, and you want to find some other function related to the cell physiology, because cell से tissue बनेगा, tissue से organ system and what. Yes, basic जो role रहेगा, that is of cell. Yes, if there is some alteration into the cell, you can easily find out if this type of alterations are present into the cell. So as a sum of these actions, we will have some effect on the human. So that's why it is considered a little bit more. It is not an easy method, but these are nowadays replaced as lot of already conventional method. For example, earlier that you have, if you study this one, skin sensitization test, skin irritation test. The person from the pharmaceutics say, okay, that if you want to evaluate any formulations, so there should be some parameters. There are some parameters. It should not be irritating. And if you want to go for the shampoos or other things, it should not have some effect on the eyes as well as on the skin. So these are some of the standardization methods. Means that you are using the rabbit skin into that. Rabbit skin is very much sensitive. Suppose rabbit skin you are using, or the rabbit or the guinea pig you are using. These are the animals which are having very sensitive skin. Suppose if you compound these having high acidic value or high basic value, means that it is having some irritant. So can you imagine that if any irritant is applied on your skin, just to find out that how is the response? Is the response? That is a mere cruelty to the animals. Just to overcome all these things, we are having this cell biometric test. For example, that we can have health and test on the chorioallantoic membrane. This is a membrane which is designed, which is developed, and we can find out that because in chorioallantoic membrane there are a number of blood vessels. So it can be used for the skin irritation studies, or you want to find out some effect on the angiogenesis as well as some irritation studies. It is quite easily used. Another one is hemolysis test or red blood cells hemolysis. Very simply that if your RBC are hemolyzed, it is having some damaging effect onto the RBC. Simply, if you want to take the RBC, you can take the blood sample from anyone. It can be from the human use, or it can be from the blood bank. Simple thing is that hemolysis, RBC can be moved, means it is having damaging effect. No need that you are going to use the animal for that, and your experiment lead to some sort of suffering into the animals. So you can avoid this one. Next one is bovine corneal obesity, or the permeability test, DBT जिसे बोलते हैं. This is one of the, you can say, very cruel tests which were used, but now it has been accepted by the OECD guidelines 437. Means earlier what we were doing that ocular irritation studies, any irritant, it was simply applied onto the ocular membrane. It was instilled into the eyes. Okay, you easily observe if you are taking a bath and some sort of shampoo or the soap is going into the eyes, how you feel like. But in this case, we know that this compound is having some acidic value, it is having some basic value, and it was instilled into the eyes of animals just to find out the behavior. And just to replace this suffering, we can have this bovine membrane, you can say corneal membrane, and it is being used. Another one is isolated rabbit or the chicken eye test. This is again a similar test. We want to evaluate some shampoo, soaps, or any irritant. That can be used into this test also. And these are some of the advantages which I'm telling you that why we are opting out for, you can say, sand-based acid. Means that is economical process. आपको सिंपल कल्चर ग्रो करना है सेल के ऊपर आप ग्रो करो and you can use the cells. Another one is that if you're growing any cell, means you will get a homogeneous colony. All the cells will be of similar type. So when similar sets of cells are there, you can isolate, you can use, and you can say multiply these cells again and again. So that is a quite easy process and easy to handle also, and moreover, the results are pretty more accurate as compared to other tests. Important thing is that when you are using the cells, there is no ethical issue, there is no moral. Nobody going to question that आपने सेल को कैसे सफर किया, आपका आरबीसी का हेमोलिसिस हो गया तो आपने क्या किया. That is one of the more advantages because you will not need any ethical permission. Is that if you are going to use any animals for your research, you need the ethical permission. Institutional ethical, animal ethical committee is there. The protocol should be passed from that. If you are able to justify that your animal will not be suffered at that extent, then only your protocol will pass. And you should be able to justify that if you are using that much number of animals, what is the use of that? Can you reduce the number? These are some of the ethical considerations which are given in any form. But in this case, if you are using this cell, tissue, or anything like that, there is no ethical consideration in that. That is a major advantage of this one. But we can say that there are certain advantages, disadvantages also. Best is that we can find the toxicity, but we cannot find the safety. Safety, there is a difference between the toxicity and the safety. Earlier, there was labs into the research, which were called as toxicity labs. But now, this toxicity is converted into the safety pharmacology. Safety pharmacology means that your compound is affecting your vital organs or not. CNS, heart, kidney, and lungs. These are considered as the vital organs. That your drug is very safe, okay, that is non-toxic. But if it is safe for these organs, so that's why, but this type of studies or this type of results cannot be predicted if you are going to use that cells into your studies. Now, next one is microorganisms. I think that everybody is quite aware of these microorganisms, and the thing is that earlier in 2000, a genome studies were used. In that case, we were studying the genome of the microorganism. School at that time, and we know the genome of that. Genome means, if you know the genome structure, means that if anything is affected, which gene is responsible for that. So that's why if we know these microorganisms are there, this can be useful for studies, as well as you can find out some other results. For example, neurodegenerative disease, Alzheimer, Parkinson, Huntington's, that can be also evaluated by this type of models. And one of the most commonly used example of this one is Saccharomyces cerevisiae. The reason behind is that we know the gene structure or the gene map of this organism very well. It is quite easy to cultivate. You can easily cultivate it, and you can easily propagate into the lab for your studies. So this is a little bit more advantageous to use these animals, these microorganisms. And again, by using these microorganisms, we can go with the propagation. Suppose that you can isolate some RNA or DNA from these organisms, and this RNA or DNA can be further amplified. There are certain techniques, methods are there. For example, PCR is here, which you can amplify the content. Means that if you are getting a very small content of RNA, DNA, that can be amplified to a lot of numbers. So means from a very small organism, you can get a lot of data by using these techniques. And these are some advantages, and these advantages associated with this one means that if you talk about microorganisms, so economically beneficial is to carry out the procedure microorganisms आपको काटेंगे, but rat or mice के case में there is a phobia among everyone when they are going first time to pharmacology lab कि rat हमें काट जाएगा तो tetanus हो जाएगा. So in that case, microorganisms will not fight to that much extent. But there is one major limitation associated with the microorganisms is that you need a biosafety lab. Biosafety layer of at least level 2 should be there because sometimes if you are using this pathogenic strains, so there are chances that it can lead to some sort of disease or uh, it can be responsible for any outbreak of that. Suppose that is the reason when we are used to the microorganisms, the care should be taken. It should be used with very professional care as well as biosafety should be considered by using these microorganisms. Next one is human tissues. As we are talking about the cells, tissue, means that this is a little bit more advanced version because you can have the human tissue. Directly after the effect evaluate करना, you can use the human tissue then, and we can have these human tissues from the cadaver also, from the donated bodies also. There's so many options are available. Surgery के बाद भी कुछ आपको tissues मिल जाते हैं. And nowadays, one more thing is that there is a reconstituted human epidermis, which is being used for this in irritation studies for the thermal toxicities. And that is a quite easy process. Yes. If we talk about this reconstituted human epidermis skin models, if you see the layers, just like that, whatever is into our skin, that is also available in that. Means that suppose you want to find out that how will be the penetration pattern of your compound into the drug or what type of effect it can produce onto your skin. So that can be easily found from this reconstituted human studies. Cosmetic के लिए irritation studies के लिए dermatotoxicity के लिए this reconstituted human skins are widely used, and that's why I told you earlier also that nowadays so many products are coming like that way that no humans or animals are used in these skins. That is the reason because they are using this reconstituted human skins, and that is also approved by the authorities. Regulatory authorities also accept the data from this reconstituted human studies. Next one is organology. That is a little bit more advanced technique, and we say that our system means if we talk about the body, we are having an integrated system. Means our cardiovascular system, we say that it is hard, but in case of cardiovascular system, our kidneys are also playing the equivalent role. In case of respiratory system, trachea is also there, lungs are also there. Means nowadays this is the technique we can say organs on a chip. Means that these are some sort of studies just like means these are basically a 3D microfluidic structure. Means it is having the exact physiological function, but your organs are behaving. So all these arrangements are present on that chip. And any change into that chip, suppose that you are injecting any compound or you are applying any compound in that, what type of changes will be there inside your body? These same changes will be affected, reflected into the chip. So that's why this is a little bit more advanced system, and it is accepted also, universally accepted. Regulatory bodies are also accepted on this. And based upon this, that we are having so many systems just like that. This one, lung origin. Can you see that? This one, this is the system. How we are having some microfluidic structure. Means after administration of any drug, absorption के बाद उसका distribution कैसे होगा, metabolism कैसे होगा, excretion कैसे होगा, same pattern will be observed in this case of studies. Again, this is heartology, this is arteriology. Can you see the structure over there into the veins, CD? Means this is the same pattern. What is happening in our body, that is also happening in this case. So what is the use to have these animals into that? can the exact results are reflected into these studies. We don't need to postulate from the animal data. Either this is the behavior into the animals, so we can have the similar behavior into the humans also. But this is just like human organ on a chip, exactly what results you are observing in case of organ on a chip. Similar pattern will be observed in case of humans also. This is placenta on a chip, this is gut on a chip, liver on a chip. So means everything is available on a chip. And now, can you see human on a chip? All the systems are placed there, all the systems are integrated. Means that if you are giving any drug, what will be the associated effect onto the other organs also? Nowadays, this is also applicable when trials are there, means that there are some certain limitations, although, but still, that is used into the advanced research labs. So you can say that some of the applications associated with this one, means drug metabolism, you can easily find out, and you can accurately mimic the lung, heart, kidney, gut, as well as any organ which you are using. Means similar results will be observed into your results also. So there are certain limitations. Means the reason behind is that for the toxicity are a little bit difficult to predict onto this case, and sometimes that in case of brain, because we know that brain is a very complicated or the complex structures. There are microcapillaries into this. But some sort, because what we are in, this is 3D fluidic structure. If there is any bubble or the air entrapment, so it can give some sort of different results. That is one of the limitations which you can say the research is going on to work on this. But still, if we are using or we are opting out for this type of method, we can avoid the use of animals into our research. Next one is in chemical method, means simply that by using the analytical models, analytical tools, for example, HPLC, you can use means that there is a Sharp's mixture. Suppose that you know that Selfish mixture is there, and if the concentration is more than 30 micrograms, that will lead to the toxicity. Means no need to give into the animals or to the human. Simply by this HPLC, if you can find out the concentration, okay, it is more than 30 microgram per ml, so means that will lead to the toxicity. Why to opt out for other experiments, whether into the humans or whether into the animals? Simply by this type of analytical tool, if you can find out that this much concentration is there, and this much concentration is responsible for some sort of adverse effect or some sort of side effects, so better to avoid this situation. So these in chemical method, I think that everyone is doing that, HPLC, HPLC, and so many other analytical tools are also there. Next one, as you can say, microdosing studies. Means that now I'm talking about another similar thing, means that animals के अलावा हम क्या कर सकते हैं? Suppose that while we give to the drug, when we start process of drug discovery, means that what we find out the toxicity as well as safety. From these preclinical studies, that we opt out for the phase 1 studies. In phase 1 studies, means we simply try to find out the pharmacokinetic of a drug. But pharmacokinetic means that those from what we are getting from the animals, that those will be calculated for the use of humans also. But if we are using that microdosing, means that हमने बहुत थोड़ा quantity का drug लिया, तो simply find out the ADME, because that ADME, just to find out that ADME, आपने थोड़ा सा भी quantity लिया, उसका process तो वही रहेगा, absorption होगा, distribution होगा, metabolism, excretion होगा. So microdosing is a little bit new concept also, that in which you can use the human molecule and very small quantity, which will not produce any sort of toxic effect that used in these studies. And the reason behind is that, suppose that we are using, you can say, animals. But as I told you, the pattern is totally different in case of humans. So in that case, I'm finishing before that. So the pattern of drug distribution is different. But if we are opting out for the microdosage, we are opting the humans in that. Human volunteers are there. And moreover, the dose what we have selected is not toxic to anyone. So by this method, we can also get good results of this. Volunteer study, microdosing as well as volunteer study. For example, you want to find out some sort of unpleasant behavior, just like addiction or pain. Because in case of pain or addiction, or suppose if nutrition studies are there, in that case, the pattern is different in case of animals, but the pattern is different in case of humans. Addiction के केस में, pain के केस में, nutrition के केस में, anything means that which is not producing any unpleasant behavior. In those type of studies, we can opt out for this volunteer studies also. Next one is this in silico studies. I think that Dr. Latter has given the presentation in detail about this one in silico. And now this is the talk of the town. If you can predict any molecule and you can predict the molecule, you can find out the receptor, you simply talk the molecule to that probability कितने percent है, exactly कितने percent आपके हैं. So the unnecessary things what we are using in case of animals, because toxicity studies and just to find out the activity of any compound, we need a lot of animals into that. So means simply that okay, that if this is a new compound and this is having interaction or this is having some docking affinity for this type of molecules, I will go for that. That if I have synthesized any compound and it is having more affinity towards the beta 1 global, why to go for this? You can say another things कि मुझे beta 3 भी करना है, beta 2 भी करना और इसके अलावा इसके CNS effects क्या हो सकते हैं? Simple thing is that okay, then it is having some affinity for the beta 1 receptor, तो cardiovascular इसका effect आएगा unnecessarily कि मुझे इसका CNS भी देखना, वो भी देखना, anything like that. Because for a new compound, we can do like this. But this in silico studies, they have reduced this procedure. Means is simply that this is the molecule, this is the line. If they are having any affinity, then we will go for this type of studies. And I don't want to go into the details of this definition of this in silico studies and all. And Dr. Latta has explained into details of this thing. And these are simple things into the in silico models. We can say computer aided molecular drug design, quantitative structure activity relationship, computer assisted learning, computer or the mathematical analysis, microfluidic chips, and glycolysis. These are some of the methods which we consider in case of in silico studies. This is molecular model. Simple है कि then आप एक molecule बनाए, molecule को synthesize उसको dock किया, उसका क्या activity means simply. These are you can say the desired model, or we can design our molecule. We know that this is the design molecule, is the structure of the molecule will be like that, and is the structure of the receptors. How much will be the activity? How much? What can be the results? What can be the consequences? Can be easily predicted out from this one. Next one, this is the procedure what we are doing in case of molecular modeling. This one is you can say suppose you have synthesized any molecules that there is an bond formation and what are the parameters, how much of the molecular size of this compound, what can be the quantum physics. And if you find all these things, then different procedures are there that if this is a known, if this is unknown, and from that we can go opt out for the STS screening, high fructose screening, and all this. But that is again a different process, and a lot of procedures are involved in that. But simply that without going into the random screening, we can simply design our molecule, we can design our research. And we can find out the results. Next one is computer drug design, and that was explained by Dr. Lan also in detail. And then this nowadays the talk of the आपने structure लिया, design किया, बनाया, खत्म किया, उसका activity check. But that is not a single process. Actually, it is a lengthy process. It's a complicated process. But the use of this method is that you can avoid or you can pass up the process of drug discovery. Conventional drug discovery की अगर हम बात करें तो that is generally a procedure of minimum 10 to 15 years. But if you're designing your molecules like that, you are restricting yourself to this area. Earlier, there's a you can say 360-degree area. But now you will find out that this is my leafy, this is my space. I have to work into this one, and there are the chances that this is the level of affinity into the molecule. So ligand can separate kitna affinity. Because without going into any random selection, we can decide, we can find out, and we can work in that area. That is the major reason that these days everybody is talking about this computer-aided drug designing. Co-sealing are like this, that easy process, and you can easily design the molecules. 3D structures of receptors are also there. These molecules are also there. Interaction can easily be verified, and you can easily predict your results. This time consequences will be there. And next one is quantitative structure activity relationship. I think every pharma student, when they are studying the medicinal chemistry, you know that if you are changing the structure, means if you are changing the functional groups and all, there will be some changes into the functions of this. As a result of which there will be some changes into the pharmacokinetics of that. There can be some pharmacodynamic changes, means that overall there can be some improvement into the process. So by simply predicting this one, that I'll change this functional group, as a result of which उसका lipophilicity change हुआ है या उसका hydrophobicity में कुछ affect हुआ, so these changes can be predicted out from this one. And this is the process how we are doing into this one QSR model. And these are some of the advantages I told you that that's a relatively faster process, easy process also. And next one is this computer assisted learning. This is what we were talking in the previous lecture, that we can design something and we can take the help of those computers or those learning. Basically, that in this case, we can have multimedia tools or just like software and all, big, where the knowledge can be utilized. And there are some of the examples. This Stopcat is there. Toxicity prediction by the computer assisted technology means that is available into the India, approved by the FDA also, simply that you can predict out the toxicity of any compound. Because if we are employing animals into the toxicity study, that's a lengthy process also. Moreover, that there are high chances that animal will die into that. By simply predicting by this topic, you can find out that your compound is toxic or not. Another one is this CD. This one XCOM. I think that you guys are using to your lab also. Means that any experiments just for the demonstration purpose. Earlier, when we used to study, we used to have these animals for the demonstration purpose. कि आपका anti-epileptic activity check करना है, analysis करना है, anything. We used to do this. But now this has been replaced because someone has generated the data. This data is placed into the software. You can simply find out if you are giving this compound, if you are giving anything to induce the epilepsy into the axis. So this will be the pattern, or this will be the course of epilepsy generation. This type of seizures will be there into the animal. But जैसे अभी स्टूडेंट बोले थे कि then हम इससे कुछ सीख नहीं पा रहे. But simply, if we go into that preview, that if you are just for the demonstration purpose, you are giving seizures to the animals, you cannot imagine the suffering of the animals. The compounds we are using for to induce the epilepsy into the animals means that in that case, epilepsy के case में हम जो evaluation की बात करें, in that case, our first parameter to evaluate is the mortality. Means there are high chances the animal will die. If it is not dying, it will have seizures, and the severity of the seizures is that much that after that animal will have long-term effect onto the behavior. Means the life of the animal is affected for a duration. and just for your demonstration purpose only, कि then आपको सिर्फ देखना है कि मुझे epilepsy animal को मैंने picrotoxin दिया उसके बाद वो कैसे behave करेगा. And you guys, can you see that अगर आपको इंजेक्शन लगाना पड़े, I mean very much happy okay कि मैं जाके इंजेक्शन लगाना. But in the lab, what we are doing? कि हमें सब अपने एक्सपेरिमेंट के लिए हमें इंट्रापेरिटोनियल देना है. Being a pharmacologist, I know that I am opposing my own profession. I am going into a different opinion. But if we talk about the humans, there's a lot of sufferings are there. आप एक लैब के अंदर 10 एनिमल दिए जाते हैं कि all of the lab 30 students they have the demonstration. Where is ki that suppose ki that every second students go for the IP registration to that animals. Then how much injection you are giving to one animal? But if you can simply predict and if you can simply learn from the computer that this is the science, this is how we give the injection. When you are going for the research, you will definitely have the use of the animals. You can learn from them. Opportunities are there. But just for this thing, you can आपको injection लगा के देखना है and all the things. That is the reason behind the PETA has banned the use of animals for the demonstration purpose. That was just the reason. Otherwise, it's not like that कि हमारे यहाँ तो हमारे कंट्री में अगर हम बात करें बहुत से एनिमल्स ऐसे हैं जो बिना देखभाल के भी मर रहे हैं. वहां पे नहीं है. लेकिन जब हम लेबोरेटरी एनिमल्स की बात करें intentionally for our selfishness we are making to suffer the animals. That is the reason behind that PETA has said that we should try to avoid the use of animals into the studies. So these are some of the benefits, you can say the heart. And next one is epidemiological studies. And these are basically the long time studies. And there was one study which is nowadays very quite easily Hangum studies. That studies continued for 70 years. 70 साल तक वो study चली. That was to study the cardiovascular disease. इस एक जनरेशन में क्या डिजीज है उसका नेक्स्ट जनरेशन में क्या है नेक्स्ट जनरेशन में क्या होगा. These type of studies are also available. Then from the epidemiological data you can find a lot of effects. For example, cigarette smoking is having a direct effect to produce the cancer. But can you produce this type of study from the QS? Can you use animals into this one? Rat, mice, dog, small animal or large animal. Can you use the cigarette smoking and cancer relationship? Can you find out in that? But in that case, lots of studies are there, epidemiological studies are there, from where the data can be utilized for your research, for your reference also. So nowadays, this is a quite important factor of epidemiological studies. That is what I'm talking about, Framingham heart disease study. So this is two types of studies are there. We can say observational studies and experimental studies. From observational studies, you can have descriptive studies and experimental studies. Just to find out, just to retrieve the data, all these studies, which can be utilized for the further research. And again, after this one is introduction to alternatives. Means 3 R's are there. Although we say that this is reduction, refinement, but these works include this one. Reduce means that you should share the data and the resource. There should be proper statistical method. There should be hybrid pharmacological or toxicological studies, complete in vitro product. Means that if you can predict the result exactly from the in vitro studies, don't need to go for the use of animals. And characterization before animal work is the structure or chemical. So you can replace the animals, large animal with the small animals, and you can refine. Refine means that's supposed to that there's no alternative state. You can use or you can decrease the suffering to the animals. So that was my take on this topic. I think I was able to make my point here. And another thing is that being a pharmacologist, I use animals in my labs, but any student who are using the animals in your lab. Yeah. Yes. Yeah. Rather mostly knowledge. Moreover, this is the LS epilepsy case planning. We can, that is a very good word. Just to release that this one. That your mind. Seven of it is also best. Even in this particular any neuro author. Incandescent is also the analytical aspect. That is, but if you find out some behavior changes, or you know some of the metabolic changes, in this case, but this is this is most of this is like metabolism or judicial myself. That is so. But neurodegenerative indicators, epilepsy, any daily indicators, that's for the particular disease. Definitely. I mean,
Thank you, sir. Thank you. So, Dr. Singh, you have really given a very good view of a new alternative testing method. Even the way the alternative to the animal experimentals, you have declared everything, and this is a new chapters for everyone. The way different kind of activity you have shown, this would 100% be beneficial for the researcher and the academicians. Thank you so much.
Thank you very much, sir. Now I would like to request our principal sir and Sudhir sir to please facilitate our speaker of today. Please, sir. Thank you very much, sir. Now I request Shri Dharmendra Singh ji and Dipendra Singh ji to please facilitate some of our research persons. Please, sir. So first is on behalf of Association of Registered Pharmacists, Dr. Vikas Sharma, for this special contribution in the field of pharmacy. He's the president of ARP. Dr. Manish Kumar. He is assistant professor. They have contributed a lot. They are contributing even for the upgradation of the pharmacy profession. Dr. Manish Kumar is assistant professor at Harper University, Patiala. He is one of the alumni of our college. And he is giving special contribution to the field of nanobiotechnology. Next is Mrs. Teamram. Is an Anika Jivra, who is the alumni of our college and is also MD of Oscar Laboratories. He's a young entrepreneur. Next is Professor Dr. Vishal Garg from Jaipur College of Pharmacy. Please, sir. Dr. Bharat Prashar, Faculty of Pharmaceutical Sciences, IPA University, Delhi. He is the director of IPA University, Delhi. Professor Dr. Sudhir Bhardwaj, is Director Divine International Group of Institutions, Gwalior. Next is Professor Pratap Singh Jadon, sir, from Divine International Group of Institutions, Gwalior. Next, Dr. Pushpendra Singh Narula, sir, from BN College of Pharmacy, Rajasthan. Next is Professor Dr. Gopal B. Giri Goenka University, Gurugram. And Professor Dr. Kali Singh Shekharaji, Principal, Mahatma Gandhi Engineering College, Jaipur.
So, thank you, Dr. Kumar, for felicitating me, but my designation was not appropriate. I wish to be part of Guru Gobind Singh College of Pharmacy for my lifetime. Please.
Next is Professor Dr. Pooja Oga, Principal, Global Research Institute of Pharmacy. Please, ma'am. Dr. Deepthi Pandita, ma'am. Dipsure, you can. Next is Mr. Sat Kumar, India HR Manager, PQE India Private Limited. They have also sponsored the cup, which you are seeing on the tables. And Mr. Gajendra Nath Tiwari, CEO and founder, GNT Case Private Limited. And Mr. Sanjeev Pandey, General Manager, Samsung Laboratories Private Limited. Then Mr. Suresh Seni, Head HR, Inswift Limited, Haryana. Dr. Varsha Tiwari, please move. Mr. Rishabh Gurnath, Associate Professor, Indus National University. Dr. Abhishek Tiwari, IIM University, Moradabad. And Dr. Ashish from NIT Kurukshetra. He is also the alumni of the college and doing great work in the field of nanotechnology. Mr. Anupam Atri. He is General Manager, Lapan Healthcare Private Limited. Thank you very much, all of you. Thank you very much, sir. Now we shall be meeting after the lunch for the second session of the second technical session. And students who are outside from our college, they will only proceed for the lunch now. After half an hour, our college will join them. All the pharmacy faculty members, please come up on the stage. Are from our college. Everyone. Okay. All the pharmacy faculty members from all the colleges, including our college, please come up on the stage.
WhatsApp का. Hey, man.
And now, please, all of you say happy birthday to Dr. Bhavna ma'am. Thank you very much to all of you. Thank you very much.
सर आप नीचे आ जाइए आप इसके साथ दांत दो का फिर सर ज्यादा बड़ा खोज नहीं आप पीछे आ मैम हाँ जी मैम नंबर दे दो अपना भी
The faculty members of Guru Gobind Singh College of Pharmacy, please be on the stage. We want to move. Come up on the stage.
सर आप भी आ जाओ प्लीज मैम आप आगे आ जाओ पीछे वाली मैम वेट करना मैम Thank you, ma'am.
नमस्कार मैडम, बहुत दिनों बाद आपसे मुलाकात हुई।
2 सालों बाद मेरा विशेष से just
तब
correct.
भी आप गोविंद सिंह जी
Back again, here again.
जीडी गोविंदा में आ जाइए।
मैंने सुना आप ही वापस आ रहे हैं.
नहीं, ये
This is what you promised a few minutes back. From the stage
वो
itself.
मैंने इनको बोला कि this is 7 acres. If they wish to develop it into a
Universe.
I will definitely go. और इस जगह से लगाव है, जो कुछ बना हो इस जगह पे।
HOD साहब से नाम कहते you were all in all here. मैं सर को एक बात कहती थी, मैंने कहा आपको काम करना आता है, करवाना पाता है कि we really still remember your management skill and leadership skill. सबकी तरफ से हर किसी की ग्रोथ की तरफ ध्यान देते हैं. बस मुझे यहाँ बिठा के आप चले गए आप.
मैं तो आपको इनवाइट कर रहा हूँ क्योंकि वहाँ के स्टूडेंट्स को बहुत नीड होता है। कम्युनिकेशन स्किल्स इज इम्पोर्टेंट पार्ट। और जीडी को इनका में वैसे ही तो बच्चा जब आता है तो पेरेंट्स का पहला प्रायोरिटी रहती है कि इसका
आपके डिपार्टमेंट में या कहीं और में? सर क्योंकि लास्ट टाइम मेरा एक्सपीरियंस वाज नॉट गुड देयर व्हेन आई अपेयर्ड फॉर दैट.
आप मेरी बात क्योंकि आप थोड़ा सा और स्ट्रेंथ आ गई है मेरे पास। मैं डीन एकेडमिक्स बन गया हूँ यूनिवर्सिटी का, कुछ मेजर काम कर रहा हूँ यूनिवर्सिटी में जानता के साथ।
Or otherwise we can collaborate online to some other platform if we can do something. DBT is now we must be in +1.
या उसको भी आप मेंटर कीजिए मैडम आप।
Sure, definitely all times. +1 at that time he was in class 6 or 7 that time. Yeah. मैम नहीं आए.
मैम वो थोड़ा सा अक्सर आती है तो फिर कमिटमेंट हो जाती है इधर उधर। मैं जिस बाऊंगा अभी तक मैं अभी एक बार फिर दोबारा हूँ। मैं तो खुद बंद करता है निकेत से मिलने का।
यानी कि जब भी बैठ जाता है ना फिर शुरू हो जाता
अच्छा फर्स्ट दिन आप दिल्ली में
दिल्ली
मैं जाता हूँ तो फिर अगर 10 दिन आएंगे तो फिर क्या है जाऊंगा बल्कि मैं अपने स्टूडेंट्स भी लेने आया
बिल्कुल लेके जाओ. और अच्छा बहुत फायदा पैनम क्लिनिक
हाँ
is flourishing by leaps and bounds.
टैलेंट था मैंने तो ज्वाइन करते ही पहचान लिया था उसको। मैंने कहा व्हाई यू आर वेस्टिंग योर
Brilliant बच्चा, exceptionally brilliant and down to earth है सबका.
पर उसे सभी टीचर्स के अलावा सबसे ज्यादा मेरी बात मान।
अभी भी बैठ जाते ना.
आपका नंबर मैम क्या है जी डबल 7 बिल्कुल मैम बिल्कुल। और गुड़गांव कभी आए तो जी बस
कब आया?
नहीं मैं पक्का
चक्कर आपका लगता ही रहता होगा यहाँ पे.
बिल्कुल
पेरेंट्स आपके यहीं पर है ना?
मदर
अच्छा
मदर से फिर ज्यादा समय दूर रहना ठीक नहीं,
ठीक भी
मतलब सारी चीज छोड़ देते हैं, मदर
वो एक नेटल बॉन्ड है ना, जो नेटल बॉन्ड है।
मिलता है। मैं ये सोचता हूँ जिंदगी में सब कुछ करके देख लिया ना, जब उनका आशीर्वाद मेरे सर पे है ना, सब चीजें हो रही। आप कहीं मंदिर में ना जाए, मैं पेरेंट्स के आगे गाने रहते हैं।
तो मोर ग्राउंडेड यू रिमेन द हायर, है ना। इस तरीके से ये होता है, जितने हम जमीन के नीचे रहते हैं ना, उतने ग्राउंडेड रहते हैं, तो उतनी हाइट हम अचीव करते हैं। वही किसी भी ट्री के साथ भी यही कुछ है।
और तो मैं मम्मी को बोलता हूँ कि तेरे को डांटने के लिए आप
सर आपने ना, अब मुझे लगता है आप जहां जहां से भी आप जुड़े हुए हो, चाहे आप अपने बच्चे से, अपनी वाइफ से, अपनी मदर से, जिस इंस्टीट्यूट से, प्रोफेशन से, तो आपका सबके साथ ही आप एक पर्सनल पर्सनल बॉन्ड है। इस तरीके से आपने हर किसी के साथ इसको, हर रिश्ते को आपने नरिश कर, हाँ बिल्कुल, वो चीज दिखाई देती है, हर जगह दिखाई काम करे। मैं मैम को यही कह रही थी, मैं क्या सबसे ज्यादा क्लैपिंग सर के ऊपर हुई। यही चीज हम लोग अर्न करते हैं, एंड इट्स नॉट दैट इजी टू अर्न। आपको बिल्कुल अपने आप को
हाँ जय मैम। हाँ
ज्वाइन करते क्या हम 24 को एक इंटरव्यू भी छुट्टी थी, छुट्टी बनाएंगे तो ये भी इसमें आप टीचर तो ये मेरे पास चलता है, कितने फ्रेंड्स में जाते हैं।
मेरी कौन खींच रही है? अच्छी खींच रखी, इतनी खतरनाक फोटो वाला चैन है। क्या कहते हैं? मेरे खा के आता है। और क्या ठीक है, ठीक है, ठीक है। कैसे आ रही, कैसे आ रही बता, कैसे आ रही, कैसे आ रही, कैसे आ रही? कैसे आ रही? ये भी हाँ ऐसे खेल। लैपटॉप में लैपटॉप में बढ़ते मुझे रेडियो लैपटॉप में हल्का सा ऊपर दे ये क्या ये ऐसे का बैठ गया चेहरे पे देखती हो ये सोफा पर नहीं है इधर देखा कुछ बहुत लग रहा है बहुत टाइम रखनी है भी जाओ
पिछले देखते हो कैसी तो अपने आप से कर सकते आगे सीढ़ी में होती अच्छा इसके बाद Initial days में jobs जाने low expenses on this stock for हर तारीख का रिस्क ना low risk है वही चलते हैं उसी जानने में रिटेल और डिस्काउंट के लिए जानने में रिस्क वही कर वही टेल वही तो लगता है कि I'm all of them
इधर दो को लेके आ, दो या तीन। आपको
Good afternoon, everyone. Good afternoon, everyone. Okay, I hope you had your lunch. What was the menu? And what was sweet dish? What was sweet dish? Okay, at least you had your lunch. Okay, yes or no? Okay, for now we start with the next segment of session. So for this, I call upon Dr. Satnam Singh, Associate Professor, AIMSM, Mahesh University, to chair the session, and Dr. Shailesh Sharma sir to co-chair the session. Please, sir. Please, sir.
Good afternoon, everyone. I said good afternoon, everyone. Actually, I believe it is very difficult to attend a lecture after lunch. The post-lunch session is very difficult for all. But the next speaker is very talented, and I hope he will make his presentation very interesting for you people. And you are requested to learn each and everything. So anyway, myself is Dr. Satnam Singh, Associate Professor, Department of Pharmacology, Asian Institute of Medical Sciences and Research, Patna, and ex-controller of examinations at Asian University. Today, I have the honor to introduce the next speaker before you. He is Dr. Rohit Dutt. Dr. Rohit Dutt is presently working as Dean Academics, GD Goenka University, and Professor and Dean, School of Medical and Allied Sciences, GD Goenka University. He is instrumental in developing new policies to improve academic structure at GD Goenka University, keeping in view of the current need of various sectors. Professionally, he is a medicinal chemist with expertise in computer-aided drug design.
He has done PhD in pharmaceutical sciences from MD University, Rohtak, and M.Pharm in pharmaceutical chemistry from Guru Jambheshwar University, Hisar. He has 16 years of teaching experience and 1.5 years of industrial experience in this University Laboratory Limited. He has more than 60 research paper publications and books to his credit so far. He had also honored with APTI Best Young Pharmacy Teacher of the Year Award in 2015 and Nirali Prakashan in 2019. And the Teaching Excellence Awards in 2021 by ASMA for the excellent contributions in the field of pharmacy education. Dr. Dutt has been awarded with prestigious silver medal by the Excellency Honorable Governor of His Excellency Honorable Governor of Haryana in recognition to his PhD research work. He is also conferred with honorary professor diploma in recognition of academic and professional excellence by Bukhara State Medical Institute, Uzbekistan, in the year 2020. Dr. Dutt was also honored with Atal Swasthya Bhushan Award 2019 by Chevron Foundation. He has delivered more than 100 guest talks on various topics of healthcare domain and organized healthcare funds in rural areas in Delhi NCR. So without taking much time, I would like to invite on the stage Dr. Rohit Dutt. Please give him a big round of applause. एक बार जोर से ताली.
So good afternoon, all. And thanks to the chairman of the session, Dr. Satnam Singh ji, for his generous introduction. And my co-chairman, Dr. Shailesh Sharma, who is a good friend of mine. It's always an honor and a difficult time to speak in front of your friends. Because rather than going into technical mindset, you always come across the memories we have spent last night. So I request all my friends to please concentrate on my presentation. Apart from the extracurricular potential, I have some technical expertise also. So, as the topic of this national seminar, national conference is on drug discovery, challenges and opportunities in drug discovery using biotechnology. So, itself, drug discovery and development is a complex process, I can say, which contains so many disciplines to collaborate together. But still, despite so many advances in medical technological field, you can say that it's very hard to develop a new drug in a span of 2 or 3 years. Still, the latest reports, which has been reported by different researchers, it takes around 12 to 15 years to develop a single drug with a $2 or $3 billion investment. So we can sense the urgency that we have seen dengue. We don't have any single tablet, we don't have any single pill to treat this infection. We have witnessed the COVID era, how we have gone through the COVID era. We are not sure that this particular vaccine, this particular pill will eradicate this virus from your lungs, from your system. So it's a great challenge to entire healthcare community. And since we are the pharmacists, and we are involved into the system, we are not highlighted as the drug developer, as a pharmacist, but we have the paramount importance. Doctor prescribe those medicines which a pharmacist developed over the years. So I think we need to understand our importance first, that we are valuable for the industry. If we are not concentrating hard, then the doctors cannot produce the healthcare system in India. So we have, before starting any session, we must have the objectives of the session. I know I have been restricted to deliver my talk for 10 minutes only, thanks to the organizers, because they have mentioned that it's a post-lunch session. So I will not trouble you much. I will take you through the historical background of drug discovery process over next few slides. And then the second objective is that there are so many computational and other techniques which are emerging over the years. I will take you through, I will rush you through those techniques also in a quick manner. Then, as this conference is sponsored by Department of Biotechnology, and I have been told that some word biotechnology should be stressed in your presentation. So I'm stressing much on the biotechnology theme. So it's my third objective, and a case study. To conclude this session, I have included a case study also, which will introduce you about the concept of precision medicine, personalized medicine. Last is the, still, I will say so much high about the techniques. I will apprise so much high about the techniques, but still there is a challenge. We need to think it over. So last slide will give you an insight where you all can think over the period, 5 days, 10 days, 6 days, and Google it through and send me the feedback, what you feel about my challenges which have been listed in the last slide. So with your permission, may I start the session? I'm just wish to get an idea that the students of which particular streams are sitting here. How many students are in the final year, B Form final year? And how many are from B Form third year? Less than third year? Lovely, they are less from. So I got an idea that the audience is, the level of the audience is much more important. Without taking through the advancement, I think I will keep it to the basic concepts, so that at this in a session of 10 minutes, you can take a leaf out of my session. That is the vision of this particular session. So if we talk about the chronicles of drug discovery, that we started from the traditional therapies in the ancient time. A brilliant example of this traditional therapy is willow bark. Willow bark has been used for the ages for treatment of the pain and the fever-like disorders. And can anyone tell me that from this willow bark, which was the bioactive constituent present in this willow bark, which is responsible for the pain relieving effect? Any student who know that willow bark was very much was cited in the literature for a particular fact, which is the bioactive constituent in that particular willow bark. So it's a question to you. Please Google it out. I will ask in the last slide in the session. Please concentrate on this, because if you don't want to listen me, at least you answer me in the last, so that you can Google it now. So there was a bioactive constituent which has been developed into aspirin over the period of time, that has been developed into aspirin. So after that, natural product science and different disciplines have grown up, and we got the techniques of bioactive fractionation, the isolation of bioactive constituents, the characterization of bioactive constituents, different analysis techniques have been developed. So we were able to isolate some of the compound, but the major problem with the herbal content, herbal bioactive constituent, that you can isolate a particular bioactive constituent over the period of time with the same quality. So that was a limitation in herbal drugs. The cognosy teacher should not mind my statement. This is a limitation that you cannot get the same quality of bioactive constituent from the herbal source every time. So that's why the role of medicinal chemistry there, whether we can synthesize this bioactive constituent into the labs, so that we can develop the same quality of that bioactive constituent times and again. So that's why medicinal chemistry has got the role, and we have synthesized the compound of bioactive constituent, which was isolated from willow bark, and then it has been derivatized as per the need of the biopharmaceuticals, whether it's an issue of bioavailability, whether it's a dissolution. So there is a role of bioavailability and other biopharmaceutical factors. So we need to derivatize the synthesized compounds. So that was the matter. This word modern is misnomer. Don't talking about so modern discovery, which was next to the synthesizing the compounds. That next, if you have an idea about the chemical registry system of chemicals, there are huge number of compounds which have been synthesized over the year. Huge number of compounds. The synthesis was going on. Organic chemists, medicinal chemists have synthesized so many compounds, but without any bioactivity. What is the use of those compounds? So scientists have thought over the years. They have come out with a solution that is there any way to reduce the number of the compounds, and then the era of computer-aided drug design has been developed over the years, and we have so many techniques which can be used as a filter before synthesizing the potential compounds. I can say if you start with 50,000 or 5 lakh for compounds and you are getting only one drug, and the surprising fact is that you are getting one drug, this drug has been asked to remove from the market after the phase 4 clinical trial, and the entire process has been lost. The entire manpower has been lost, and the company is on the company has, because the big companies have invested substantial. You can see that 12 to 15 years of research and $1 billion investment, and if still molecule fails in the market in phase 4 clinical trial, I think it's a totally discouragement for the fact in the industry also, as well as the scientists who are working on that particular concept. So that's why there is a need that any tool or technique that can increase this efficiency of drug discovery process. So scientists have deliberated after that. Is there any technique that we can have a handful of molecules to synthesize, which shows there are potential of being a bioactive constituent? So I can say this, that QSA, it has been reported by one of the European Union and recommended by REACH also, US FDA also, that the precise use of in silico technology could reduce the overall cost of drug development to 50%. So means any process which can reduce the cost and the time. Cost and the time is of paramount importance for the pharmaceutical industry, and QSAR is the simple techniques. So the thing, everybody has heard the name of QSAR. And anybody who can tell me anything about QSAR online, 4th year student, please stand in. Any student, whatever you know about QSAR. I'm not asking any technical aspect, I'm not asking anything. Just tell me what is QSAR.
It's a qualitative structure as an relationship in which we deal the top drug on the basis of their chemical structure.
Okay. Hardly right. It's not a qualitative, it's a quantitative. It's a quantitative activity structure relationship. So you have told that you are relating something. So let me come to the next slide. Obviously, the reason behind this QSA, the compounds with known biological activity has been fed to the computer, and you are getting new compounds with improved biological activity. It means something prediction is going on. Means QSAR model is telling you something about the substitution of the compound, the substitution of different parameters, what should be the chromophore of the molecules. So this is the output we are getting. You can read the number of compounds which are required to synthesize a compound. अगर आप benzene की 4 position पे, if you put different groups, then how many compounds you are going to have? So are you going to synthesize all those compounds? It's a waste of time. So it's always better, rather than going for each and every derivative, synthesize a small number of compounds as an output of QSAR. But there is a problem in quantitative structure activity relationship. We can say SAR. There is a problem in SAR. On the one hand, you have structure, image, and on the other hand, you have a numerical value in terms of biological properties, AC50 value, IC50 value. So how you are going to correlate an image with a number? So this is a problem which has been solved by QSAR. Anybody has any idea about this? That how you can relate a number on the right hand side with an image on the left hand side, which is the molecular structure? Is there any way out? There is an image and there is a number. You want to correlate the series. There are 20 images on the left hand side. There are 20 numbers on the right hand side. And I am asking you to correlate both these.
We can give the numbers.
You can give some numbers. Very good. You can give some number, but how to convert image into a number? That is my question. Because if both the terms are numerical, you can compare them. There are a wealth of statistical techniques reported in the literature, but how you can correlate? How you are going to convert a structure into a number? That is the main thing, and this has you can convert this image into a number through the use of molecular descriptors. As the name suggests, it's a description of a molecule. You are describing a molecule into a number. I will give you an idea. Suppose you have 20 compounds on the left hand side. You have chemical structure. You have the right hand side. You have IC50 value. If I say that each compound has a refractive index, every compound has a molecular weight, molecular refractivity, log P value. Yes, please try. I was looking for the student who can say that it's a Lipinski rule of 5, that you have certain fixed parameters which represent a compound. So you can cite those parameters as molecular descriptor, as in numerical value. Now on the left hand side and on the right hand side you have numerical values, and now you can correlate. You can just put a graph, select both the cells in Excel sheet and make a graph, standard graph. You have the thing you have done in your analysis and other subjects also. Just make a graph and calculate R square value. R square value will give you an equation, and in that equation you can just put your value of the unknown compound you want to synthesize, and that equation will predict the value of biological activity. After this prediction, if you want to synthesize hundreds compound, just put those value in that particular equation and you will get the value of biological activity, and filter those compounds which are coming into your range of biological activity and synthesize those compounds. So this is a basic concept of converting an image or chemical structure into a numerical value, and numerical value can correlate it. So there are on record, there are 356 correlation techniques, whether it is a linear regression or whether it is a classification, whether it is 2D QSAR, whether it is 3D QSAR. Now it has reached to 7D QSAR. So there are so many techniques which you can use on these two values if you both have the numerical values. So I think it's clear to all that how the image can be converted into numerical value. So there are different kind of descriptors in QSAR study which are related to constitutional topological. Topological means 2D descriptors, which don't consider any hydrogen in the molecules. 3D descriptor means which consider hydrogen molecule, which consider different aspects of different, you can say, coordinates along the X, Y, and Z axis. That is 3D descriptor, 3D QSAR. If you are not using it, if you are using a hydrogen depressed molecular graph, it means it's a 2D QSAR. There is a simple difference between 2D and 3D QSAR. And amalgamation of both the techniques are needed. I am not here to criticize one technique because every technique has its own limitation and application. So we need to use a conducive approach, a consultative approach, where we are integrating both 2D and 3D into our mode of building. So that is, first you filter your compound through 2D QSAR, then apply 3D QSAR, and you will get, suppose you have started with 10,000 compounds, it may reach to 10 compounds. And synthesize those compounds and play to board, then you will get the predictive value. So there is a book by Roberto Torresini and Diana Consoli. They are the co-author of my work also during my PhD. So they have written a very comprehensive, very good manual of molecular descriptors. 3,500 molecular descriptors have been reported in this book. Their equations have been reported. Lipinski rule of 5 has only 5 parameters, but you have 3,500 parameters which can represent a particular molecule. So you can use those descriptors and you can go through the equation itself. Luckily, I don't want to praise myself. 22 molecular descriptors developed by me, my guide, has been incorporated. Again, it's for students and maybe for staff also, although my guide has restricted me not to disclose the real calculation part, but I'm not obeying my advice of my guide today because I've reached to a level where I wish to transfer my legacy of my guide to the audience itself, the young population. It's always better to disseminate the information as much as you can possible, and I think conferences like this is a real platform to disseminate such information. So I will give you a simple example where you can easily understand, you can calculate at your own level how to convert a structure into a value, into a formula. So there you can see structure 3 isomers of pentane. The first one is normal pentane, isopentane, and third one is new pentane. So you put numbering to the carbons arbitrarily at your own wish. You can start from anywhere. Just put the number 1, 2, 3, 4, 5. Then in the 3 structures, then first is the distance matrix. You need to create a distance matrix. I need to conclude it in 5 minutes. Thank you. So I will quickly complete in 5 minutes. Yes, I want to describe this slide to the students. Yes. Yeah. Yes. Thanks a lot. Distance matrix means you need to write the number of carbon in a compound on the x-axis, number of carbon in a compound on the y-axis. Then just calculate the distance of 1 to 1. From carbon number 1 to carbon number 1. What is the distance? 0. You put 0. From carbon number 1 to 2. What is the distance? 1. Do it in the similar manner. You will get a value of distance is SI. Calculate all the vertices. These are known as vertices. And total the whole value. What is the first value? What is the whole sum of that good distance? 6, 7, 10. You think it's 17, 23, 40. The first value is 40 and second? 9, 15, 6. 8, 8, 7, 39, 32. Yeah. So from 40 to 38 and 38 to 32. So what you are getting? When the compound was just like this in normal phase, the distance was more. When the distance was less, the value is less. Then again in the last structure, the value is again lower. Then which distance has a relation with the structures. The distance can discriminate 3 structures or maybe 30 structures. So you can calculate values of 30 structures and put it on the left hand side, and on the right hand side you can put your biological activity and develop this correlation. Next example, adjacency. Adjacency means who is sitting next to you. You need to consider that value, who is sitting next to you, that's all. So from 1 to 1, it's always 0. From 1 to 2, from 1 to 3, there is no between them. Just calculate this. And again, what is the value if you can calculate? 12 and next. It's 16. So it's 12, it's 14, and then it's 16. Means 12, 14, and 16. Reverse. The molecules are closer in this matrix. More the closer molecule, more is the value of that particular molecular descriptor. So there are 3,500 such descriptors which can discriminate the structure, and you need to develop these. Okay. So it's a simple formula. This is known as one of the finest and the first molecular descriptor developed by Wiener in 1947 that calculate the value half of the sum of distance matrix, which I have shown it in the last slide. So 40 was the value for first, and its half is 20. So this is the value of Wiener index. Wiener index can be considered as a molecular descriptor. I request Dr. Bharat Pasha to have a cup of tea. 36 by 2, 18. And 32 by 2 is 16. Why this? Out of mind, if you have any idea, why the value has been done in half? We are looking for a compound of 5 carbons, and you have seen the structure of the drug. How many carbons? So many, more than 40. What will be the value? It will be a 2 lakh, 3 lakhs, 4 lakhs. So normalize the value to a readable point. We have done it 1 by 2, and you can develop a compound. You can put it 1 by 4 also, and it will be a new descriptor. And you can publish this data, and you can get a Scopus index publication. So look for those molecular descriptors. And let's say clapping of recognition, or just I need to write a session. Of course. So has been reversed in this whole. Here we are saying that molecular descriptors cannot be related to biological activity. Now this problem has been solved. We have joined hands, and now you can correlate these activity and moleculars. So this is the basic concept I can share in this call in the time frame which has been given to me. And I will take it to you the last slide rather than going into details. This is the book for students. For students, that this software is free. This software is free on the website by the name of eDragon. Please, what you need to do if you want to calculate molecular descriptors 3,500 in one go, in 1 minute, just use this software. What you need to input is your molecular structure in mole file. Molecular structure in mole file. And what is this mole file? If you have a licensed version of Camtra, you can sign it as a mole file, save it as a mole file. Otherwise, India में तो सब कुछ crack हो ही जाता है. Otherwise, you have the crack version with you, save it into mole file, put those structures into this software, load descriptors, and you will get 3,500 values. What you need to do, you need to see whether how to choose those descriptors. Next question, that out of 3,500, which are relevant ones, how we can analyze this. So a simple concept to rationalize the utility of descriptor is that check for the duplicacy and check for the degeneracy of the value. If a molecular descriptor list is giving the same value for different compound, it means it is of no use for modeling. We need different value for different structures, unique values for different structures. Use those descriptors. Out of 3,500, you may arrive to 100 descriptors, develop the equation. And if you get maximum prediction rate more than 90%, use that model. So it's a simple QSA technique. So there are different techniques which you can use through this software itself. It's free. Whatever you want to use, it's free on eDragon software. I have shown you a simple graph that compound 8, we don't know the value of compound 8. You know the value of compound 7 is the same chromophore, and you want to predict that what will be the biological activity. So what I did, I have prepared the model file, and we calculate the values of different descriptors. What is the result? You can see. Calculated log EC50 is 1.60. Now I will synthesize this compound, and after synthesizing this compound, I will assess the value. If it is coming near to 1.55 to 1.65, it means my model has a good prediction rate. So this is the way to do out. There are different models. I will skip it. This slide is very important with the respect of this, the theme of this conference. It is more important to know what sort of person has a disease rather than the disease person has. Again, read it very carefully. It is more important to know what sort of person has the disease rather than the disease a person has. It means our old concept to start the drug discovery from a disease is changing. There is a paradigm shift in this drug discovery process. Now you need to think what kind of a person has the disease. Like my chairman and co-chairman, they both are different. Their genetics are different. So how a drug molecule can elicit a same response? Dr. Suresh Sharma may be benefiting from 500 mg of dose, but Dr. Satnam may not get the same response in 500 mg. Dr. Shailesh can get the hypersensitivity reaction. On the other side, Dr. Satnam may not get it. So we need to think right now where requirement of the drug pharma industry is that you need to think for a person for the genetics. The genetic makeup of that particular person who is approaching to a doctor for the treatment itself, the doctor need help now of the genetics. That's why the theme of this conference is very relevant: drug discovery in development using biotechnology. So now you need to think about genetics. And luckily, I'm telling you, as an expert of computer-aided drug design, that this genetics can be predicted. This genetics can be predicted. Your family history is a molecular descriptor for us. Your height over the year of your family members is a molecular descriptor to us. Your diabetic profile over the years is a molecular descriptor to us. We can predict that what kind of disease, what kind of genetic makeup this person has, and we can reduce it. So I will conclude it here with this mindset that you can use biotechnology techniques. There are so many biotechnology techniques. I have prepared so many slides, but it has been wasted by the organizer of this conference. So slide you can see that mix of the picture above represent the old, you can say modern drug discovery, but you need to look into this, the bottom line emojis, which is that this population contains some the drug which are safe to somebody, drug which are not safe to another one. So we cannot elicit a same dose or drug to any same population of mind. So you need to think about, apart from disease, that is important, but you need to think about the person itself. That is the role of bioethics. So there are, I will skip these slides. And this has been proved. This has been proved. We have substantial reports that a particular drug is eliciting hypersensitivity in one genetic makeup and producing good effect in another genetic makeup. You can go through these through your literature exam. So this will be the, I think. Hello. I think this will be the new age chemist shop, new age retail shop, where you are going for your medicine and they will ask you for your genetic sequence. On the basis of genetic sequence, you are getting this. Thank you once again.
Thank you very much for your very solid question. You have technology. You have used the involvement of the audience in your lecture. You gave the very nice and simple real examples to learn these things. And you gave a concept of the green chemistry to our audience. Sir, thank you very much. आपने शुरू किया था एक्स्ट्रा करिकुलर एक्टिविटी से इस स्टेज पर आकर अच्छा लेक्चर देने के लिए जरूरी है कि आपके पास पहले एक्स्ट्रा करिकुलर एक्टिविटीज भी हो, वो आपको कॉन्फिडेंस देती है और वो आपके आज में झलक देती है कि एक्स्ट्रा करिकुलर एक्टिविटीज में यू आर एक्सीलेंट, साइंटिफिकली यू आर एक्सीलेंट और ऑडियंस को बांधने में भी यू आर एक्सीलेंट. थैंक यू वेरी मच सर फॉर योर वैल्यूएबल टॉक विद अस.
चलिए जी, देखिए नेता बनने के बाद ये हुनर तो आई है. Luckily, with the support of all my students, all my faculty colleagues at Guru Gobind Singh College of Pharmacy, I was successful in getting a post of National Vice President in APTI. And just I want to represent my community on the larger front. So, from the desk of this conference, I wish to thank each and everybody for supporting me. Keep supporting me. I will be your voice in the times to come. And thank you.
Well, and I will thank you to my co-chairman, Dr. Satnam Singh sir, for a nice introduction of our guest. So thank you, Satnam Singh sir, for your precious time. Thank you once again.
Thank you very much, sir. Now I request LA sir, please, and our principal sir to felicitate Dr. Rohit sir, please. Sir, I also request you to please felicitate Satnam sir also. Ms. Rupam Sharma from Indus International University. Please, ma'am. Ms. Poonam from Indus International University. Mr. Ashok Kumar from IC5. Mr. Akshay Parihar from IC5.